everyone here, even if you are just little names on a screen. My name is Jacey Bell, and I've been interested in mushrooms for a very long time. I'm old enough that I went to the U of S when there were still booth labs offered, and so each biology student needed to take all the 200 level courses. So I did a little bit of self-study work on mushrooms in the lab and just became fascinated by them. I am currently working as a lab instructor at the U of S, and I'm learning right now to switch over to all remote teaching. So this is my first time giving a webinar. I hope that it goes well and we don't have any technical difficulties. I do have Luke here. I don't know if his video's on, but he's got audio. If you want to say hi, Luke.
Hello, everybody.
He is going to be moderating the chat because I find it kind of distracting to go from my notes to the PowerPoint and then back to the chat. So hopefully he'll catch all your questions and he can stop me and I can answer them, or he might even be able to answer them for you via text. I do have you guys all automatically muted when you come in to the webinar, and that is just because I find that sometimes Zoom has that infinite echo if we all have our audio on, our microphones on. But feel free to unmute yourself if you want to ask a question, or if we miss you in the chat or something like that. Let's see, what else should I tell you? I am recording this, so just a heads up, if you are typing in the chat or have video, then you might end up getting recorded, or I guess if you use the audio. As long as that doesn't bug you, it's no problem. I think the chat actually doesn't get recorded now that I think of it, so if you're shy about having your voice recorded, the chat's the best way to go. Let's see here. So on the PowerPoint up there at the beginning, for those of you who haven't been hanging out in the waiting room for a bit, you can see I've got all the Saskatchewan Mycological Working Group information on there, and then my email address as well. And you're welcome to send any questions anytime to those. So I guess we'll go ahead and get started. All right, so the Saskatchewan Mycological Working Group is a really new group in the province. It sort of spawned out of the need for data regarding fungal species in the province. A few years back, I was at the Alberta Mycological Society's Great Alberta Mushroom Foray, which is amazing, and I highly recommend you guys to go to that as well if you're really interested in mushrooms. But we had a presentation by a biologist who was checking out all the species occurrences across the Western provinces. And as it turns out, there's very, very little data for what occurs in Saskatchewan. So the goals of the Mycological Working Group are to collect species occurrence data for the province and just to generally educate people about local fungus and I guess fungi in general. So some of our members have indicated interest in learning about fermentations like sourdough and things like that, because yeasts are a fungus. We want to look at identifying mushrooms, maybe photography of specimens, all kinds of things. So if there's anything you're interested in regarding mushrooms, let us know and hopefully we can make something happen. Oh, I know mushroom cultivation has been another really big topic we've been talking about. You'll notice over here, I have in stars, we need a logo. So if anyone's good at graphic design and, you know, wants to donate a lovely logo to our working group, that would be great. I just wanted to give a shout out to the Native Plant Society of Saskatchewan. They have kind of adopted us so that we have a larger established group to work under. I am currently the treasurer of the Native Plant Society of Saskatchewan, which I guess kind of by default makes me the treasurer for the Saskatchewan Mycological Working Group as well. yeah, so feel free to contact us there anytime, join up. We'd love to have more people interested in mushrooms. A quick summary here of what we're gonna go over today. I'll just give a quick introduction to fungi in general, talk a little bit about their structure, their ecology, and then we're going to get into important features for identification. So we'll look at where it's growing, what it looks like, and a little bit more of the, I guess, technical spore prints, things like that. And then I'll show you some collecting equipment. You really don't need too much for fungus, and some resources. If we have time at the end, I can also go on to explain some fungal biology in more detail or do a Q&A. Totally up to you guys. This can be as interactive as you want. Like I say, I'm just learning about the remote teaching. I'm used to being face-to-face with my students, so yeah, feel free to ask as many questions as you like. So to begin with, the basics of fungi. They are composed of hyphae. They're basically little thread-like structures that are within the substrate, or sometimes growing on top. I've seen some very cool photographs where bark has been peeled away from trees with the mycelium crawling all along it. I believe Sandy added a photo to our mycological working group Facebook page, which is really good. So the cell walls are made of chitin. The reason that this is important is chitin is a really sort of like tough structure. It's indigestible to humans, and it plays a part in the way that fungi get their nutrition. So these guys can't move. They're not like animals, right? Our fungus is just growing on a substrate, in a substrate, throughout the soil, that sort of thing. And it doesn't have chloroplasts, so it can't make its own food, right? Which means it's a heterotroph and needs to eat other organisms. And if you guys have been in intro biology courses, you'll see that like amoeba will engulf their prey and that sort of thing. But because of these hard cell walls that fungi have, they're unable to do that. So they digest their food extracellularly, which is actually really cool. So they'll secrete enzymes into whatever substrate they're on. Some of them are more generalist. Some of them are incredibly specific and only eat certain things. So where a fungus is growing is really important for its identification. But it basically breaks down, sort of liquefies the substrate around it and then absorbs all of the nutrition. Yeah, I think that's all I'm going to say for that one. We will move on to the next slide here, if my computer will work for me. There we go. So that brings us to the roles in the ecosystem. Because of its extracellular digestion, fungi is an incredibly good recycler of carbon in the environment. So it'll break down anything dead, basically, sometimes living things, some fungi are pathogens as well. It breaks down the detritus, so the fact that we have fungi is probably a reason for less forest fires, to be honest, because if we have all this downed wood and nothing was recycling it, it would build up over time and probably our forests would burn more often. As well as dung. A lot of fungi grow on, you'll see on like cow patties out in the pasture and that kind of thing. And it breaks down that waste as well and allows the carbon and the nutrients to be used by other organisms. And our last point there, sometimes fungus, especially mushrooms, will provide food for other organisms. I'm sure all of us have seen squirrels carrying dried mushrooms to hide in your shed at the cabin and things like that. Oh, I almost missed on here, form associations with plants. So there's a couple of really neat ways that fungi interact with plants. They can form mycorrhizal associations, which is essentially penetrating the outer tissue of a plant's roots And it allows communication with the plant and I guess photosynthates from the plant. So the sugars that the plant is producing is sometimes passed to the fungus. The fungus might give phosphorus in exchange to the plant, or it might pass water and that sort of things as well. Additionally, there are some very cool plants that do not photosynthesize. So I have a picture of one that's commonly called Indian pipe here. It's a really beautiful plant and it has no chloroplast. It doesn't make its own food, but it has an association with fungi that allows it to get nutrition. Additionally, if any of you have been to the Cypress Hills and have seen pine drops, it's the coolest plant. It's a bright pink color, and it actually feeds off the photosynthates from lodgepole pine. And it's basically like a fungal bridge is formed between the two plants, the lodgepole pine and your pine drops, and they're able to pass sugars back and forth that way. So it's a really, really neat way for plants to be connected to each other. All right, so we've gone over what is fungi, right? Now, more specifically, what is a mushroom? Because I think that's what all of us are here to talk about is the mushrooms that we see around the province, right? So a mushroom is a small part of a fungus, essentially. It's just the fruiting body. So I've got some images here. This is our typical mushroom here. And as I mentioned, it's made out of hyphae, so it's very tightly packed hyphae. And up here on the gills is where the spores are formed. And this part of the fungus will grow either when the conditions are right or maybe when the mycelium is under stress. So underground, the part that we don't see is the mycelium, the grouping of hyphae, right? So here I have a picture of a jar with some grain in it, and it's completely covered by hyphae, this white cottony growth. So you have all this fungus, but no mushroom. This is actually one of the starting parts of cultivating edible mushrooms, which I can always talk about at another time, but unfortunately don't have time for today. And then this picture here, I was fortunate enough to get some fungus growing on paper towel inside a plastic container and was able to kind of show, like, the mushroom here is what we see above ground in the forest, right? But there's all of these little hyphae going on underground. So the hyphae, the mycelium, is persistent, whereas the mushroom is short-lived and just for spore dispersal. Oh, there we go. And I see someone is having trouble getting in here in the waiting room. There we go. I think that worked. Apologies for the delay there. So for those of you who were here early, I passed on a link with some mushroom terminology. And I'm just going to see if I can get that into the chat again. Fungi. TML. There we go. I thought I had it copy pasted, but in any case, if you go to that website there, it's also here on the PowerPoint. It's an awesome glossary. This is a website, an old one actually, that's hosted by the University of Saskatchewan. And once you get into fungal identification, you're going to notice your field guides. Depending on the publication, it'll have a full ton of terminology. You can have a terminology overload in some cases. So this glossary with pictures I have found immensely helpful in navigating all the terminology. I'm going to go over a couple of the basic ones here. So on our general mushroom, we'll have a cap, although of course not all of them have it. You might see the term pileus used. I think that generally isn't used in regards to the outside of the cap, but you will see it used interchangeably in some cases. Then there's the gills, of course, which not all fungi have, because there's an exception to every rule, right? The gills are also sometimes called lamellae, so just note that people use that interchangeably as well. Some of them will have an annulus, which is a little ring around the top, and what that is the remnants of the inner veil of the mushroom. So as the mushroom's coming up out of the substrate, it's folded in and has a protective veil, oftentimes covering the gills, which tears apart. You will see this on your like cremini mushrooms that you get from the grocery store. Then there's the stalk, the part that elevates the cap above the soil, also called the stipe interchangeably. And then the last one that you should know is vulva. And this is the remnants of a universal veil. Now, not all mushrooms have this, but you can correct me if I'm wrong, but I think some of the more poisonous mushrooms do have that. So it's a good idea when you're collecting, if you dig down under the soil a little ways and kind of pop the mushroom out and see if it has that vulva, that can help you to identify what you're looking at. So the function of mushrooms themselves is all about spore dispersal, right? Proliferation of the species. So if you look at the structure of a mushroom, and we'll be seeing a bunch of different shapes here, you can kind of think about how the shape of the mushroom helps the spores be dispersed throughout the environment. So remember, fungi can't move, so they have to use everything else at their disposal to move their spores. So your general sort of basic mushroom with its gills and its stalk, it just elevates those hyphae above the soil, and the gills are shaped in such a way that air currents can easily blow the spores, which are formed on the surface of the gills, and move them to other areas. When you have the bracket fungi on trees, it's the bottom part that is the fertile layer. So if you look really closely, you usually see little pores, tons and tons of little pores, and lining the inside of those pores is where the spores are formed. So they kind of trickle out the bottom of that mushroom. And again, growing way up in trees, those spores can catch the air current really well. So hopefully, if that spore's lucky, it's gonna land somewhere where there's a good food source for it and germinate. Now this is one of my favorites. I don't know if any of you have seen these before. They're called earth stars. They're so cool. They're basically like a puffball shape, and the spores are inside of this sac. Now when it comes up out of the ground, this thick outer layer is surrounding that inner layer, and it pushes out of the earth, and as it dries, it splits open. And then once this is mature, the spores become all dry, and the whole unit will detach from the ground, and the wind can actually blow this and tumbles and sort of lets the spores out as it goes. So these are some of my favorite. I try and keep dried specimens around when I'm teaching labs to let my students play with them. Sometimes I think it's more for me than them, just because it's super fun. And oh, I see a question here. We do have earth stars here. This photograph is from Hinton, Alberta. But I've definitely seen some, oh, gosh, I can't remember now. But they definitely occur in Saskatchewan. The next sort of dispersal method I'll talk about is autodigestion. So that means self-digestion. This is really cool. I absolutely love these mushrooms. They're the inky caps, and this one I believe is a shaggy mane inky cap. These are actually 3 different mushrooms, but the same at the very least, the same genus and with the same type of spore dispersal. So early on, you can see it's a nice looking mushroom. And I've cut it open here so you can see the long lines of gills inside, and that's where all the spores are produced. Over time, as they mature, these guys will start to turn dark, and you can see that's happening here, and that's because it's digesting itself, essentially. So as these mushrooms become digested, the cap tends to curl outwards and upwards, and the spores will kind of drip down in inky black drops, hence the term inky caps. And this is perfect for wind or water dispersal. So the wind can blow them once they're dry, water will run off and drip the spores away. This last picture here, generally they don't just lay down like this. This is from my backyard. It's an inky cap that I really wanted to photograph going through the process, but my rabbit had other plans and decided to crush the mushroom. So I took a picture of it the next day and it had mostly digested itself. You can see it's really inky here still, but there's a big puff of dark black spores, really dry there. So they kind of blew around the backyard. Okay, here is 2 different species of puffball. These are another favorite of mine just because they're so much fun. So we have Lycoperdon on the left here. And these guys have a tendency to stay mostly closed, but when they dry up, they get a little hole in the top there. And you know what? I'm gonna actually show you a specimen that I've got here. Oh, in the chat I see there, shaggy mane equals delicious. I have not yet tried them, but I hear before they start digesting, I'll just go back a slide, when they're still nice like this and all white, they're supposed to be very good cooked. But depending on the species, you're not supposed to have them with alcohol, because in some people it can cause sort of a toxic effect and make you ill. But, oh, can I tell you if mushrooms are edible or not as I'm talking about them? Yes, if I know, I will let you know. In some cases, I know if they're edible, and some of them I'm really not sure. But if you guys know if they're edible, definitely drop me a message in the chat and let me know. I've only just started with foraging for edible mushrooms, and I'm always excited to know myself if they are edible. All right. I'm just gonna get out of the screen sharing here. So hopefully, do you guys just see me now? Big screen? Okay. So I've got this dried puffball here I've been saving. If you're ever out in the forest and you see these little Lycoperdon guys dried up like this, it's really fun if you put your phone camera to slow motion and do a video of poking it with a stick. So the spore dispersal mechanism on this one works really well with raindrops. When a raindrop hits, here, see if you guys can see this, you get a puff of spores coming out. I'll do that one more time. Super fun, right? And from what I understand, most, if not all, puffballs are edible, but only at a very young stage. There, I'll go back to my screen share. Hopefully you guys can see the PowerPoint again. The one on the right here is a Calvatia species, and I'll tell you right now, my Latin is not good. I was never able to get into Latin courses at the U of S because they conflicted with others, other courses I was taking. So I just wing it with the Latin pronunciation, but it's all good, I guess. So this one, actually, you see it a lot in cow pastures. They're very, very big round puffballs, and at maturity, they dry up and the top breaks open. So wind and rain can disperse these spores, and if you look, it's almost like a large cup just filled with that brown dust. So you can imagine how many spores are being produced there. Just give me a second here, and I'm gonna see if I can find the chat again, because I can't see you guys anymore. There we go. Yes, I hope I don't have mushrooms growing in my house. I do have a lot of specimens in here, though. I don't think it's damp enough for them to grow. All right, I'm gonna get into some major identification features, things that you're gonna wanna watch for when you're out documenting the species that you're seeing. So first of all, where is it growing? The really neat thing is that a lot of mushrooms are or fungi are very specialized, so they'll only form associations with certain kinds of trees. Maybe some of them will only grow on hardwood. Some of them will form associations with the roots of conifers and that sort of thing. So it's a very good idea to check out basically what trees they're growing under. If you're in like a hardwood stand or something, and you're keying out your mushroom later, and it comes down to two species, and one of them has an association with, say, pine trees, you'll know it's probably not that one. Oh, I have a couple of questions here. So one of them, as far as I know, all puffballs are edible. I don't know if you should quote me on that one though, because I haven't tried them all, but I know that a lot of them are. If any of you guys know, feel free to drop that in the chat. I know that a lot of puffballs are definitely edible. Oh, and the morel in the picture have a relationship with a certain tree. Yeah, usually morels grow in association with aspen or hardwood trees. I've found them, every time I've found them, they've been close to either an aspen stand or I found them in Manitoba in an oak forest and that kind of thing. Let's see here. Oh, that is an excellent point, Jamie. Yeah, so the young destroying angel amanitas are ones that have that universal veil that splits open to be the vulva later on. So if you're collecting and you find something white and round, it is a good idea. Do a longitudinal section and see if it's got those gills forming inside, or if it's just puffball, so just white all the way through. That's a very good idea. And is the other one Trametes versicolor? I'm not actually sure. That is an old photo and I can't remember where it was even taken. It's entirely possible though. So yeah, so these ones, take note of where it's growing. The general shape of mushrooms is important too. That can often get you started in a key. So we've got little puffballs here, so they're nice and round. These ones here are growing upright and have gills on the underside. We have the bird's nest fungus. I absolutely love these little guys. They often grow in mulch, in gardens and that kind of thing. We have ones that might not have a stipe. They're mostly just like a cap and gills. We have the cup fungi that have their spores on the inside. And then there are also the coral-shaped ones. There's even more. There's kind of like an infinite different shapes of mushrooms, but these are some of the basic ones that can help you get started in an identification key.
Jacey, someone asked if Saskatchewan has any known poisonous mushrooms.
Yeah, But we have a few, a lot of them can be up for debate. I've seen a lot of debates on mushroom forums about whether or not, say, like verpas are edible or toxic and that kind of thing. We do have amanitas that are quite poisonous, so I'd be careful with those. Yeah, assume it's poisonous unless you know it's edible. Lisa, I think that is a good, kind of a good way to approach it. I never eat anything that I pick unless either I'm with someone who knows a lot better than I do. I'm not lying with the shirt. I am still an amateur mycologist. Or even if I know that something is edible. I found my first morels this year, and I know they're edible, but I don't know how I'm gonna react to them. I have a slide later on that says eat a little bit the first time, make sure it's really well cooked, see how you react to it, even if you know it's an edible one, just in case. It's like an allergy test kind of thing. The other very important thing to look for that's sometimes overlooked by people who are just learning to identify mushrooms is what's underneath the cap. Does it have gills? Does it have pores? Does it have teeth? Sometimes two mushrooms will look very similar, but one has gills and maybe one has pores. This is really cool. These, sorry, I'm losing my train of thought here. The mushrooms with teeth on the underside always surprise me. I go to lift it, turn it over, and I don't know, I'm delighted every time that it's got teeth instead of gills or pores. In any case, all three of those are the spore-bearing structures. Looking at color and bruising can be extremely helpful. I love this photo of this purple mushroom. Unfortunately, this one's from a trip to BC. I haven't found anything quite this vibrant in Saskatchewan yet, except for a couple of fly agarics that were nice bright yellow. You can see one of them down here in the corner. And then there's this guy in the middle, kind of a rusty orange. In some cases, it's a bright orange. This one's a Lactarius deliciosus, which I have never eaten, but I hear they're not as delicious as the name implies. But essentially this one, when you touch it or cut it or break it, it bruises like a deep sort of olive green color. It's kind of startling at first, and it looks really gross sometimes when you see an older one. But the color that a mushroom bruises can be indicative of what species it is. The other thing to look at is the texture of the cap and the texture of the stipe, or stalk. I never did get a chance to identify this mushroom, but its sliminess was absolutely intriguing. And there are certain species that are known for their sliminess. There's one called a slippery jack, I believe, that's often found under pine trees, and it's got this weird slimy texture to it. This one's lecithin, so it looks like a bolete. It's got little pores on the underside of the gill, but these scales on the stalk are what give it away. So it's a sort of like black scaliness. These are ones I just recently had a discussion about with people about it being edible. So a lot of people eat these and call them red caps, I believe. The odd person will have a reaction to it though. So be careful if you're gonna eat these red caps. This one is the fly agaric, and the little white dots all over the scales on the cap are one of its identifying features. The Mario mushroom, if you will. And then here's another one that has a really neat pattern of scales on the top. So all of these are good identifying features. Additionally, if you've ever seen a russula, they kind of just look like a plain white mushroom. Often they've got a red cap on them. They've got gills. They don't seem super special until you feel the way they break. They snap like a piece of chalk. It's the coolest texture. So if you think you're in a patch of russulas and you take one and throw it at a tree and it just kind of explodes into little pieces, then you've probably got a russula.
Somebody wanted to know what the purple mushroom was in the last slide.
Oh gosh, I did identify it once. I don't remember what it's called, but I will get back to you on that one. I'll actually make a note. Purple mushroom here. Thanks, Luke. I'm glad you guys are asking questions so I can kind of direct where I'm talking. I can talk about mushrooms for hours, honestly. My students know this from the mycology labs, but I'm trying to keep on task here. So yeah, if there's anything you're interested in and want me to talk more about, feel free. So scent and taste can also be important when you are identifying a mushroom. Oh yeah, I could put the names on the slides and get back to you, although it is recording. I will find a way to get any that I know how to identify. Some of them are just photos I've collected over the years, but if I know the names of any, I can find a way to send those to you or even post them on our Facebook page. So an example of scent here is this guy. It's called strawberries and cream, or sometimes the bleeding tooth fungus. Bleeding because it has this little bright red secondary metabolite that it exudes. And you'd think it's fairly distinctive. It's pink. It's got little teeth underneath the cap. If you're unsure and you want to make really sure what it is, if you smell it, it has a really nice sweet scent to it. So that's a good indication. And then taste. I know we've been going on about how you shouldn't eat mushrooms if you don't know what they are. However, in most cases, if you put mushroom in your mouth, taste it, and spit it back out, you're good to go. There are some keys that use taste as a defining feature of mushrooms. So when I was just first learning to identify them a few years ago, I couldn't figure out this one Lactarius. I didn't know what species it was. I knew the genus, and somewhere along the key it asked about a spicy flavor. And one of the people who was leading the foray asked, well, did you taste it? And I thought, is that safe? But apparently you put a little piece on your tongue, take it out, and if it's incredibly, incredibly spicy, then you have a red hot Lactarius. So that kind of delighted me when I found out that you could narrow down your mushroom ID by taste. One more way to help yourself identify the mushrooms is to do spore prints. So I didn't have great photos of this. I need to take some. I've been trying not to use any photos that aren't my own or friends so that I can record this and send it out to you guys. So there's a kind of blurry picture down here at the bottom, but you can see I've got two plates here, and one of them has a bunch of little colorful marks, and the other one has little chunks of mushrooms. To do a spore print, just take a piece of the cap, or the whole cap if you want, put it down on a surface. I personally prefer to use glass if possible, because when the spores come out, if they're a light color, you can put them on a dark background, or if they're a dark color, put them on a light background, and then you'll be able to see the color of the spores, which can be a defining feature. I highly recommend putting like a coffee mug or a cup over top of it if you're going to leave it overnight. Not only so the air currents don't blow your spores around, but also because when you collect mushrooms, more often than not, they've got some sort of insects or maggots in them. And I know from experience, if you leave out a bunch that you're identifying on your kitchen table overnight, you might wake up with a lot of maggots on your table in the morning, which, I don't know, not my favorite thing to clean up first thing in the morning. So that's spore prints. Oh, you can also do spore galleries, which is kind of a fun thing if you've got glass. We've done this in the lab with my students with microscope slides. You make a spore print on a microscope slide and then put another one on top, seal it around the edges, you can save that. Those spores will be viable for a long time, dried out like that. So you can put them under the microscope later if you want to look at the shape. You might be able to germinate them on agar or that sort of thing if you want to grow them out. There we go. So collecting specimens for identification. Some people take their field books out with them, and I admire the patience they have to sit out there and identify things in the woods, in the field. I prefer to collect the mushrooms and take them back and identify them in the evening wherever I'm staying. Sometimes people worry about picking the mushrooms being a bad thing, but honestly, if you're chucking them back into the forest or the pasture or wherever you happen to be when you're done with them, you're actually just helping them disperse their spores. So it's a good thing. Stuff that I bring along includes a basket. So I've got my favorite basket here, this big one. Couple bucks at Value Village. It had the added benefit of holding favors at my wedding, so it's been used for lots of stuff. Paper bags are a really great idea. Mushrooms don't do well in plastic or anything they're gonna sweat in when you're carrying them around for the day. And I mean, you can throw everything just into your basket, but I like to tag each specimen with a number or a note of where I found it and put it in its own paper bag, just so I have that information for later when I'm going to identify it. Which brings me to note cards or a notebook. Just to make notes on what you see, what is it growing with, anything that might be of interest. I should give a little shout out to the Alberta Mycological Society here. When I went on their Great Alberta Mushroom Foray, they had a really cool system of pre-printed little note cards where you could just circle things like the habitat that it occurred in and things like that. And you could photograph that with your mushroom or put it in the bag with it. And it was a great help later when we were identifying. A knife is totally optional if you want to dig out the mushroom. Some people find it unpleasant to dig down with their fingernails, of course, to see if there's that vulva at the bottom. And then a camera or smartphone. I love to take pictures of the mushrooms in the habitat that they're in. I always find this helpful later on. And then the smartphone brings me to my next slide here, if my PowerPoint will work. Sorry, guys. I'm just going to give you guys a really quick crash course on iNaturalist. I don't know if you guys have used this before. I absolutely love this app. And it's being used by the Saskatchewan Conservation Data Center, the SKCDC, to log species occurrences. So this is the easiest way to help out with observations of different fungi in the province. So on your smartphone, if you download iNaturalist, click on More in the bottom right here, and then scroll down a little bit, and you should be able to search for a project. If you search for Saskatchewan Conservation Data Center, this is a screenshot of my phone, so it's already got that on here. There, you can just ignore that. We'll go to the next slide here. You'll find the project, click on it, and you'll go to the project page, and then click join. Again, on mine it says leave, but that's because I'm already a part of the project. So if you click join there and follow the prompts through, there will be a place that asks about location data. So make sure that you click on yes, no matter who adds the observation to the project, and that way your location data will be shared with the data center. And essentially that can drop little pins on a map of where different species are found around the province. So it'll really help out with making species lists for the areas. Yeah, I guess the other thing is you'll have to turn on location while I'm using the app for iNaturalist. I made the mistake of doing an entire BioBlitz with my location data turned off, and I had to manually put pins on the map for where I found each species. So just a heads up, great idea to turn location services on if you're going to use iNaturalist. The other great thing about it is it sometimes suggests what species you might be looking at. Even if you don't know what it is, you can still log it, and other users are able to go in and say what they think it is that you've found as well. So we can get a consensus between citizen scientists as to what species we think we're seeing. Oh, here's just my little disclaimer on eating foraged mushrooms. I know we talked about this one already. I just, yeah, just make sure that you're really careful. Different people have different reactions to things. But for the most part, as long as you're careful, you can definitely enjoy foraging mushrooms in Saskatchewan. I was pretty psyched finding as many morels as I did this year, and I'm planning on teaching my puppy to sniff them out next year. I don't know if he will take to it, but I think that would be a really fun activity for us. Go searching for mushrooms together. The last thing I want to tell you guys about is different resources that I find useful. There's a ton out there, and you might have other ones. Feel free to drop it in the chat if you have some really favorite resources. I'd always love to hear about more. But I've added that USask Fungi of Saskatchewan website. As of right now, their list is basically what we have for species occurrences in Saskatchewan. So the more we can add to that, the better. And I've mentioned the Alberta Mycological Society quite a bit, so I've added them to the list. I guess they're the only other mycological society I have been involved with, and I think they're doing a great job. So when we started up this group, I've been modeling it a little bit after what they do, as well as putting our own spin on things. Yeah, Geo, absolutely, you can pop up on video.
Oops, start video. Let's hope this works. This will be good for your thing here. Cool. Okay, hopefully this shows well enough. I'll probably get in there. It's really kind of bad, but what I'm trying to show you here, it's mushroom identificator app. So it's like edible, not edible, and so like that's just the basics. But it has same thing you're talking about. You can put where you found the mushroom. There's a community, so you can help people identify, or you can help identification with yours. But one of the best things I found and how I learned most of what I know is it has this. So it's asking me which one's dead man, or what do we have now? Scarlet, who, I don't know, I think this might be this one. Oh, hell yeah. Sorry. So yeah, it's just like, if you get it right, it's like, oh yeah, you get it right. And like, you can like continue to try to like name. So I'm pretty sure it's a tinder fungus. Yeah. Funnel chanterelle, this one. So it's just like, yeah, it's a really cool and neat way to learn identification without like, really studying it.
Oh, that's fun. It's like a little, an app with a quiz in it, basically.
Yeah, and then like, yeah, picture. And I'm assuming like the ones they use here are like very common, and like they know after however many people viewing it, like this is the thing you're looking at. Yeah. Well,
and like, so
cool. For me, it's kind of bad, 'cause, like they have like the general names sometimes, like turkey tail, but like all these are all the Latin scientific ones, and that's the ones I have trouble learning, because like, oh, here's like, it introduces it to you as turkey tail, but then it's goes into Trimedes versicolor there, and I'm just like, ah. Anyways, I just wanted to quickly show that. Thank you, and thank you for running this group and stuff. Like I said in the comments here, I've spent time in BC, and like I'm a part of the. This is Eric, by the way, he'll be coming too. I spent time in the Vancouver Mycological Society. I helped them run their show actually last year.
Oh, cool.
So it was interesting when I came here, and like I know there's a small group here, like Jeffrey, he joined the group recently, and Rena, Moss Mama, like she sells like mushroom logs and does classes and stuff.
Mm-hmm.
But they didn't know you guys existed. Yeah. So anyway, glad I found you.
we didn't really like truly exist until like this spring.
Oh, hell yeah, cool.
It's been in the works for a little while, but it
Yeah, cool.
I'm glad to find so many people who are interested, though. This is really great.
Yeah, there's more than you think. Like I said, now that you guys like have a group and like have something to like, you know, come around, like they'll come. We build it, they come.
I hope so.
Yeah, cool. Thank you so much.
Yeah, no problem. Another one I wanted to show you guys here is All That the Rain Promises and More. David Aurora is hilarious and for some reason likes to pose with his musical instruments, but this little pocket guide, it's like, I don't know, 20 bucks or something, and super awesome. It is a good companion to his bigger book, which is Mushrooms Demystified. Also love this one. This one here, one of my favorite textbooks, The Fifth Kingdom by Bryce Kendrick. There's a couple different editions of it. The older one is in a typeset font, which is really neat. This is a newer one. I've used it with my students before, and it's got a lot of good information. It gets pretty in-depth. That'll explain different types of fruiting structures and ascomycetes and that kind of thing. But yeah, this is a lovely one. This one, it makes me very sad. It's out of print now, and it's really hard to find. But if you can ever find Mushrooms of Western Canada, it is a beautiful illustrated book. Lots of nice little color pictures here. Occasionally you can find it on like Amazon or eBay or something, but you have to pay too much money for it. But anyway, secondhand bookstores, if you can find that, you're golden. And the last one I wanted to show you guys, it is for children, the Mushroom Fan Club, but I like to say it's for adults too. I don't have kids and I own it and I love it. It's got cute little faces all over, but it actually is really, like really educational. It talks about like the morale there. And of course the puffball. So anyway, that's by Elise Gravel. I want to get this for all of my friends' children. That makes me happy. Right. Let's see here. Oh, it's exactly 8 o'clock. I tried to keep it to an hour here, so we're doing pretty good for time. But if anyone's got any questions or if you want to pop up on video and share anything, you're welcome to do so. Otherwise, thank you so much for attending. I couldn't believe how many people signed up for this. I hope I was able to give you some good information in such a short crash course. I could literally talk about fungi for hours. So yeah, hopefully you've got a little bit more knowledge to start identifying your own fungi. And thanks for coming. And I'll stick around for a few more minutes if you've got any questions or comments.